Interpreting the hepatitis B markers has always been overwhelming, with all these confusing charts and endless markers, and I remember sitting in your seat, staring at these markers and trying to piece it all together. But today we are going to break it down and simplify their interpretation, and by the end of this video, you will be able to confidently analyze these markers, which will help you distinguish the different phases of infection, whether it's acute, chronic, resolved, vaccination, or even occult infections. And along the way, we will explore what happens during each infection phase, clarify the principles of treatment, and discuss how these treatments work. So let's make sense out of it all together. But first, we need to answer a very important question. Does it really matter that we accurately identify the patient's infection phase? After all, we will give all the infected individuals treatment. Well, that's not quite the case. Since not all patients infected with the hepatitis B virus require immediate treatment, but the treatment decision is carefully based on the patient's comorbidities and on identifying the infection phase the patient is in now, which is based on the markers present, as these markers will give us insight into the activity of the virus and the liver damage. So we need to accurately identify them to avoid unnecessary treatment or even worse, missing the chance to intervene at the proper time, which could seriously affect the patient's outcomes. To do this and avoid drowning into the marker's dilemma, let's approach it differently. I will take you on a journey into the world of the hepatitis B virus, where we will explore its structure and see what happens as the virus sneaks into the body and tries to take over, and how the immune system responds. It was just another normal, peaceful day inside the body, where the liver, that quiet powerhouse, was minding its own business, processing nutrients, filtering toxins, producing bile, and keeping everything in balance. But then the invader arrives, which sneaks into the body through contact of the hepatitis B infected blood or body fluids, as in case of sharing needles or unsterile medical procedures, tattooing, piercings, needle stick injuries in the healthcare setting, through sexual contact from an infected partner, or through vertical transmission, which is the transfer of the virus from the mother to her baby, especially in regions where the hepatitis B virus is endemic. And once the virus gains access and enters the body, this small but mighty invader is not just a random force, but this arrogant virus says, I'm engineered for this, and I will do my best to evade the immune system and persist in this body. So let's have a quick zoom in on our invader to see its machinery. First, we can see the outer viral envelope, which is a lipid bilayer, studded with proteins called the hepatitis B surface antigen, which comes in 3 different sizes. This envelope helps the virus survive the bloodstream, and upon arrival at its target, which is the liver, it uses it to attach to the liver cell and facilitates its entry. Moreover, it has a role in distracting the immune system, which we call immune evasion, and it can also induce an immune response, and these will be discussed in few minutes. Next, we can see the viral nucleocapsid, which is a protein shell made of protein called hepatitis B core antigen. This shell encapsulates the viral genetic material, which provides stability and protection to this genetic material. At the heart of the virus, we can see its genetic material, which is a partially double-stranded DNA, and attached to it is the DNA polymerase enzyme, which works its magic and is responsible for the viral replication, as we will see later. And finally, this is the hepatitis B E antigen, which is a secretory protein derived from the pre-core protein, and this doesn't have a structural role, but it is produced during the active viral replication, and so it's used as a marker indicating high viral replication. In addition, it can evade the immune system and induce tolerance, as we will see later. Now that we are familiar with the viral structure and how it enters the body, you need to know that this wicked virus doesn't just wander aimlessly in the body, but it has a well-designed plan to reach its target, which is the liver cell, And once it arrives to the liver cell, it uses its hepatitis B surface antigen to attach and enter it through a receptor called the NTCP, which stands for the sodium torturate co-transporting polypeptide. This receptor is responsible for the bile acid absorption, but in case of the hepatitis B infection, it has another job, which is inviting the virus in. So think of it that the NTCP receptor is the cell gate, and the virus uses the hepatitis B surface antigen part like a key to unlock it. And like a skilled thief, it enters the cell by endocytosis. And once inside, the virus takes off its protective outer coat, which we call uncoating, where its envelope is broken down and the nucleocapsid is released into the cell. This nucleocapsid, as we mentioned earlier, encases the viral DNA, but this DNA is not ready to start the replication process just yet. First, the virus needs to make its way into the nucleus, which is the cell's control center, and here it undergoes a crucial transformation where the gaps in the partially double stranded DNA are filled to make a more stable, complete form called the covalently closed circular DNA. Think of the covalently closed circular DNA as the virus blueprint, where it acts as a mini chromosome and serves as a template for all the viral activities within the cell. By this time, as you can see, the cell's own machinery is hijacked by the covalently closed circular DNA, and it starts producing several transcripts, each with a unique role. Among them is the pre-genomic RNA, which is translated into the viral polymerase enzyme, and the core proteins that form the viral capsid. In addition, it serves as a template for the reverse transcription, as we will see in a few seconds, to form the new viral DNA inside the nucleocapsid. Next are the pre-sRNAs, which encode for the different sized surface proteins which are formed and processed at the endoplasmic reticulum Golgi apparatus complex to be later involved in the formation of the viral envelope of the newly formed virus. Moving next is the pre-core RNA that forms the pre-core proteins, which is processed to form the hepatitis B E antigen, and this, as we mentioned earlier, doesn't contribute to the viral structure, but it is a soluble protein secreted in the bloodstream during active replication, which helps the virus evade the immune system. Next is the xRNA, which translates into the X protein, an incredibly important protein in the virus machinery, playing a key role in the virus life cycle as it interacts with the host liver cell, affecting its behavior. But to fully understand it, we need to go through the rest of the virus life cycle and the immune system against it to see how it influences the disease progression and the viral persistence. After this wicked virus succeeded to turn the host liver cell machinery to its own benefit to serve for the formation of its own proteins, the story gets even more exciting in the cytoplasm, where the viral polymerase and the core proteins are produced. The core proteins assemble into a capsid and combine with the polymerase enzyme and the pregenomic RNA to form the nucleocapsid. And inside this nucleocapsid, the polymerase enzyme works its magic and uses the pregenomic RNA as a template and converts it back into DNA in a process called reverse transcription. And so we end up with a new viral particle where the DNA is enclosed by the nucleocapsid, and this particle can take two routes. Some return to the nucleus to maintain an increase of the covalently closed circular DNA reservoir, allowing the infection to persist, while some head to the endoplasmic reticulum, where they are wrapped around with the hepatitis B surface antigen, forming a complete viron which is packaged into vesicles and released into the bloodstream by exocytosis, known as the Dane particles, to infect new liver cells. Before we move on from the viral life cycle, I need you to know that the hepatitis B surface antigen production far exceeds the amounts needed to wrap the newly formed viral particles. This excess amount is secreted into the bloodstream, acting as a decoy, meaning that these antigens distract the immune system, wasting its resources on clearing the free floating antigens rather than attacking the infected hepatocyte or the virus itself. Sounds like a clever strategy, right? Another thing to know is that the hepatitis B surface antigen appears 2 to 10 weeks after exposure to the virus, with an average of 1 month after exposure, meaning that it appears even before the onset of symptoms. In addition, it's present during the phases when the virus is not replicating, and this continuous production, despite the fact the virus is not replicating, is a result of the viral DNA integration into the host liver cell DNA. It's like the liver cell is reprogrammed to keep making the surface antigen as long as the virus is present, making the hepatitis B surface antigen a very important marker for all phases of infection, and its persistence more than 6 months denotes chronic infection. Another marker to remind you of is the hepatitis B E antigen, which is only secreted during the active viral replication and it indicates high infectivity. And like the hepatitis B surface antigen, it acts as a decoy. In addition, it can also suppress the immune system, promoting tolerance, allowing the viral replication without being attacked by the immune system. And this will be discussed in few minutes. You see, a really wicked virus. But as I review the viral life cycle, and I think you would agree with me, that apart from being wicked, it was a very peaceful virus that didn't cause any liver cell injury, or in other words, it's not a cytopathic virus. But it appears to be a self loving guest that is simply focused on one thing, making more and more copies of itself, but without causing any liver cell injury. So we are not really facing any troubles, right? Well, here's the twist. While the virus was busy loving itself and replicating, it was triggering an immune response to get rid of the invader. And so the severity of the liver damage and the progression of this infection is not caused directly by the virus, but rather on how the immune system responds to the virus. And so it's a battle between the immune system and the virus, and the sequelae of this infection is based on the outcome of this battle. So we need to know what happens during this battle. After the virus invades the liver cells and starts its replication process, the immune system launches a multi layered attack, and the outcome of this battle depends on the strength, timing, and coordination of the immune response. In a healthy person with intact immune response, it starts with the first line of defense, which is the innate immune system, which uses its secret weapon, the interferons. This starts when the innate immune sensors, which include the dendritic cells and the macrophages, detect the viral components like the hepatitis B surface antigen, hepatitis B core antigen, and the viral DNA, which activate the pattern recognition receptors and the toll like receptors. And this sounds the alarm and releases the interferons. And these tiny molecular messengers arrange a complex strategy designed to slow down the viral replication, where they act as alarm signals, producing antiviral proteins that slam the brakes on the hepatitis B virus replication, slowing things down and buying time for the immune system to mobilize its forces. They can also activate the natural killer cells, unleashing a search and kill mission against the infected hepatocytes and so helping in the viral clearance. They can also supercharge the immune response, activating the macrophages and the dendritic cells and so enhancing the antigen presentation to mark the infected liver cells for the cytotoxic T cell snipers to target and kill them. Moreover, it creates a protective barrier around the infected hepatocytes, limiting the viral spread while keeping the surrounding tissues calm and inflammation low. A very smart strategy from the immune system to contain the enemy without a collateral damage. And so, this natural interferon response in the acute hepatitis B infection will keep the virus in check until all the immune responses are fully activated, and this coordinated effort will help the adaptive immune system clear the virus within months from infection. Because despite the efforts of the innate immune system, it's not enough to fully clear the virus on its own. And here the adaptive immune system steps in. Starting with the cytotoxic T cells, these cells are the most important soldiers in the battle against the hepatitis B virus. They are trained soldiers that have a specific mission to target the hepatitis B infected liver cells and kill them. But you need to know that these soldiers are not just after the virus, but they eliminate the entire host liver cell. Why? Because this liver cell, as we mentioned earlier, is now a virus factory with the presence of the covalently closed circular DNA integrated in the cell's DNA and serving as a persistent template for producing the viral particles. And so, this tough but smart strategy will help the immune system eliminate the infection. And while this immune attack is the key to clear the infection, it comes at a cost, as this hepatocyte destruction will trigger the liver inflammation, and that's why the liver enzymes rise as a reflection of the immune system attack against the infected hepatocytes. Moving next to the T helper cells, the commanders of the immune response. Although they don't do the fighting themselves, but they guide and amplify the attack by releasing their cytokines, which activate the cytotoxic T cells to destroy the infected hepatocytes and stimulate the B cells to unleash a wave of antibodies against the virus. And this coordination is essential for controlling and potentially clearing the infection. And now let's talk about the antibodies produced after the B cell stimulation. These antibodies are more than just molecules, they are storytellers, narrating the immune system victories, struggles, and the enduring efforts in their battle against the hepatitis B virus. And now let's start with the anti hepatitis B surface antibodies, or in other words, antibodies against the surface antigen part. These are the victorious flags of the immune system, making the end of a successful battle. Imagine that you have antibodies that can bind to the hepatitis B surface antigen part and neutralize it, meaning that the virus will not be able to attach or enter the cell at the first place. And so, if these antibodies are detected in the blood, it's a sign of immunity, a proof that the body has built defenses strong enough to repel the virus if it ever tries to return. And now that we understand how the hepatitis B surface antibodies work in providing lifetime immunity, I believe you will appreciate the logic behind the hepatitis B vaccine mechanism. This is a recombinant form of the hepatitis B surface antigen introduced into the body, and since it's a harmless antigen as it doesn't carry the virus or cause infection, it will serve as a safe way to stimulate the immune system and produce the hepatitis B surface antibodies just as it would during a natural infection. And once these antibodies are generated, they remain in the circulation, ready to neutralize the virus upon future exposure. In this way, the vaccine prepares the immune system in advance, ensuring that if the virus ever enters the body, it will be immediately recognized and eliminated before it can establish the infection. And so, this means that if we detect anti hepatitis B surface antibodies in the circulation, it indicates lifetime immunity to the virus after recovery from an acute infection or after successful vaccination. And now let's talk about the hepatitis B anticore antibodies. Unlike the hepatitis B surface antibodies, they don't neutralize the virus and they are only produced after a person has been exposed to the actual virus, not the vaccine. Think of it as a scar on the soldier's face. Its presence denotes that this immune system has fought this virus before, whether they have won or lost the battle, and so it is present in both current and resolved infections. Moreover, they can tell us whether the infection is recent or from the past, acting as a timeline marker of the infection, where in recent or ongoing infections, we can detect the IgM anticore antibodies, and over time, the IgM levels decline and is replaced by the IgG anticore antibodies, which remain in the circulation, serving as a lifetime marker of past infection. And so, if we want to differentiate between a person who has positive hepatitis B surface antibodies, whether it's due to a previous resolved infection or vaccination, you should order the anti-hepatitis B core antibodies. Moving next to the anti-hepatitis B E antibodies, we previously mentioned that the hepatitis B E antigen denotes active viral replication and high infectivity, and so the presence of antibodies against them denotes shift in the infection dynamics, saying that the virus has been tamed to some degree and the infectivity is reduced. And so it's a message from the immune system saying that we have gained some ground, but it doesn't say that the battle is over like the hepatitis B surface antibodies. It's rather a marker of transition from an active to an inactive phase. And now, since we're familiar with the viral life cycle and the immune response against it, let's put the virus journey on this graph where we'll illustrate the rise and fall of different markers during the acute hepatitis B infection. And if you get to know how to read it, you will decode the entire story from this graph. At the start of the infection, we can detect the viral DNA since the virus is replicating. And then we can see the hepatitis B surface antigen rising around 4 weeks from exposure to the virus. This is the first clear marker announcing the viral presence. But around 12 to 16 weeks, we can see that it starts to decline as the immune system responds. And soon after the appearance of the hepatitis B surface antigen, we can see the hepatitis B E antigen appearing, reflecting that the virus is replicating rapidly and is highly contagious. And as the infection peaks, the patient starts experiencing the hepatitis symptoms as the immune system is fighting back. This immune system will start producing the antibodies that we mentioned earlier, and the first antibody to appear is the IgM anticore antibodies, denoting recent infection. And over time, the IgM levels decline and is replaced with the IgG anticore antibodies, which persist for life, regardless the fate of infection, whether the patient achieves recovery or chronicity, serving as a lifetime marker. Do you remember the scar on the soldier's face? And as the time passes, the hepatitis B E antigen fades and is replaced with the anti-hepatitis B E antibodies, showing that the viral replication has been tamed. And as the immune system gains control and clears the hepatitis B surface antigen and causes their disappearance from the blood, the body finally produces the anti-hepatitis B surface antibodies, the victorious flags and the ultimate marker of recovery and immunity, meaning that the body has won the fight and is now protected against future infections. And this is the happy ending for the battle in patients with intact immunity. Before we move on from the acute hepatitis B infection, imagine that a patient walks into your clinic and you suspect acute hepatitis B infection, and you order the usual two commonly tested screening markers, the hepatitis B surface antigen and the hepatitis B surface antibody. And now imagine that you order them during this phase. You will be definitely deceived. As you can see here, there is a time gap where the hepatitis B surface antigen, the hallmark of the hepatitis B infection, has disappeared as the immune system begins to clear the virus. Meanwhile, the anti-hepatitis B surface antibodies hasn't yet appeared, and this is what we call the window phase. This is a very tricky and deceptive phase, as you might be tricked into thinking that the infection is not there at all, but hidden beneath the negative hepatitis B surface antigen and antibody lies the only clue, which is the IgM anticore antibodies. Miss this and you will fail to diagnose your case. So next time you suspect acute hepatitis B infection, remember the window phase, this blind spot, and please order the IgM anticore antibodies as part of your workup, as this marker holds the answer. And now that we've covered the acute hepatitis B infection, it's time to test your knowledge. I will display a few questions for you to answer. After that, feel free to take a break and come back when you're ready for part 2. In the next video, we'll dive into the chronic hepatitis B infection, the available treatment options, and how we can manage this condition effectively. See you soon in part 2.